So what I'd like to talk to you about a little bit now is what happens in eukaryotes after transcription. Before we get into the cytosol and the cytoplasm to perform translation, there is a step called RNA processing. And I just want to talk briefly about what goes into this particular process. So, we've made an initial or primary transcript. Sometimes it's called pre-mRNA. So it's going to have a 5' end. And then we're going to have several different things here. We're going to have a start codon somewhere in here. I'll draw in green. And then we're also going to have these various regions which we either call exons or introns. I'm going to draw the introns as purple, the exons as red. Okay, this will be the last exon. And then I'm also going to have a sequence down here as well, towards the 3' end. So again, right here, this is exon 1, exon 2, and exon 3. And then I have intron 1 and intron 2. Now there's several different things that have to happen with this primary transcript before we're ready to move into the cytoplasm and begin translation. The first thing that's going to happen is we're actually going to take this primary transcript and we're going to attach things to the two ends. So on the 5' end of this transcript, we're going to attach a modified guanine, 3 phosphates, that's called a 5' cap. Okay. Then we're going to have a region here that I drew up here called the 5' UTR, which stands for untranslated region. We're going to have our start codon again. And then as well, we're going to have our exons and our introns. Go ahead and draw those in. Now over here in the 3' end, we're also going to have what's known as a 3' UTR, untranslated region. And in this region, this is where we have that transcribed polyadenylation signal that I spoke about in the transcription video. And this polyadenylation signal is going to result in us placing a poly-A tail onto this molecule. Call it poly-A tail because there are multiple adenines. That is the poly-A tail. Now, there are several different functions of the cap and tail. These both apparently serve the purpose of facilitating the movement of this molecule from the nucleus to the cytoplasm, because right now we're still in the nucleus. Additionally, once out in the cytoplasm, the cap and tail appear to protect this primary transcript from hydrolytic enzymes. Furthermore, the 5' cap appears to be utilized when the ribosome attaches initially to this transcript. So, we've gone from this primary transcript to this modified capped and tailed transcript, but we're not done yet. Recall that in class, we talked about the spliceosome and those SNRPs that are ultimately used to excise introns. So we're going to go through this modification or this processing where these introns are essentially cut out. So we cut out those 2 introns, and at the end of all of this, what that's going to mean is we're going to have, I'm just going to draw it shorthand now, our 5' cap, our poly-A tail, and then we're going to have our 3 exons running one next to each other, the other, and then our 5' and our 3' UTRs. So this is our mature mRNA molecule. This is what is going to go to the cytoplasm and is going to be translated at the site of the ribosome. Remember, only this region here between the start codon and the stop codon will actually be translated. So this is our translated region. But everything else here will remain on this mRNA as it's moved from the nucleus to the cytoplasm. And also remember, in the lecture, okay, I talked about the idea that we could do alternative RNA splicing where we cut out this whole region with exon 2 included in those 2 introns, and then we would just get an mRNA molecule that contained exon 1 and 3. So when we go to splice, we can sometimes get rearrangements of these exons or removal of some of these exons. And that's okay. If you lose exons, it's all right. But one thing that we don't see happen in our mature transcript is we don't see the inclusion of those introns. So this is RNA processing. So after transcription in the nucleus of eukaryotic cells, we're going to process the RNA and then send it out to the cytoplasm so that we may then translate it. So that's it.